So just a clarification, I'm not working only at the Vancouver Prostate Centre, but I'm also working as medical oncologist at the BC Cancer in Vancouver. So I wear a double hat. I'm a scientist and I am a medical oncologist. So I'm what we call a clinician scientist, because I do have my own lab in the Vancouver Prostate Centre where we do basic research in prostate cancer, but I also see patients in my clinical practice at the BC Cancer. So what I'm going to present today is why it's important to have a clinician scientist like me, and how I can help to fill the gap between basic research, so what we do at the Prostate Centre, and the clinical applications, so what we actually use to treat our patients with prostate cancer every day in the clinical practice. So there is a gap between these two worlds, and I know that for many people this is not like quite immediate to get, but in a lot of centers in the world, there is, like, for prostate cancer specifically, there are researchers who works in the lab and do like very basic research. So they work with molecules, with DNA, with proteins, but they don't really communicate with clinicians. So it's sometimes difficult to translate what they discovered into the clinical practice. And on the other end, there are many centers in the world where we see patients with prostate cancer, but there is not really the good environment to translate clinical evidence or clinical data back to the lab to try to understand why certain things happen. And by certain things, I mean, for example, why certain patients respond better to some drugs and other patients do not. So what the clinician scientists do is try to fill this gap and create more communications between these worlds, because that's the only way we can actually make progress that are clinically meaningful for our patients. So I really do both. So I have my own lab at the Prostate Centre where we use cell from prostate cancer patients and test new drugs, but I also see patients with our GU Biobank, which is a provincial biobank where we collect blood and tissue. We can go back to the bench and do some analysis in the tumor tissue or in the blood, try to understand the biology of prostate cancer. So you all know, I think, that prostate cancer is really a sport team, because we have many professionals, medical professionals involved, and the urologists usually do surgical treatment, so when the cancer is localized, and then there is a medical oncologist like me who treat patients with medication, so that can be hormone therapy, chemotherapy, PARP inhibitors, immunotherapy. So the choice and the options are increasing day by day. And then there is the radiation oncologists who treat prostate cancer with radiation therapy. And usually we can combine or integrate these treatments. So the medical oncologists, essentially, as I mentioned, treat patients with medication. And I'm very excited to be part of this extremely dynamic and promising field of prostate cancer treatment, because as you can see here, it has been a true revolution in terms of treatment options for our patients with prostate cancer. So when I started my residency in 2008, the only thing that we had was ADT for patients with prostate cancer. That didn't matter if they were castrate sensitive, castrate resistant. In 2007, there was the first trial with docetaxel. So it took like probably 1 year to have docetaxel available in metastatic CRPC patients, so castrate resistant patients. And then like in 2013, I think we started to understand that if we used chemotherapy, docetaxel, earlier for patients with castrate-sensitive disease, the overall survival, so the lifespan of our patients was going to be affected and significantly prolonged. So this is what we started to do in 2013, 2014. And then all these new drugs targeting the androgen receptor came on board, first for mCRPC patients, so castrate-resistant patients, and then later on for castrate-sensitive patients. And then like something even better and more in line with the precision medicine, so personalized medicine happened. So in 2017, this new drug that's called pembrolizumab, it's an immunotherapy drug, was approved by FDA, the Food and Drug Administration, which is the US drug agency that approved drugs, was approved not only for one specific tumor site, but was approved for any patients, including prostate cancer patients, with this genomic alteration in the in some of the DNA, some of the genes that control DNA repair. So patients with MSI, microsatellite instability, could be treated with pembrolizumab, at least in the United States. And then, as I mentioned, all these new drugs were used earlier on, and we had new drugs in a certain specific subpopulation of patients with prostate cancer, who are patients with the castration-resistant disease, but without metastasis, so M0 CRPC. And this is now standard of care, so we can use this medication for our patients. And lately, in 2020, we had the phase 3 trial with olaparib, which is an oral medication. It's a PARP inhibitor that has been approved also by Health Canada for patients with prostate cancer with DNA repair gene alteration. So this is a drug that was used for ovarian cancer and for breast cancer patients if there were these alteration, and it's now available also for patients with prostate cancer with this kind of genetic alteration. And lately, like just like few weeks ago, ASCO, that is our big meeting for medical oncologists, the data about lutetium PSMA treatment were presented, and the study showed a significant advantage in terms of survival in patients heavily pretreated for metastatic castration-resistant prostate cancer. So this also is coming on board. So there are like plenty of treatment options for our patients with prostate cancer. What we also do at the BC Cancer in Vancouver and makes this center special or more special compared to other places is we do a lot of research. And as I mentioned, lab work research, but also clinical trials. So in Vancouver, our clinical trial unit has currently 18 active prostate cancer trials. And we do have this British Columbia Provincial Biobank Research Project where more than 1,000 patients with prostate cancer have been enrolled. So this is a very precious source of tissue, blood, and clinical information for future research projects. And this is again like summarizing the treatment options that we have in prostate cancer. We still have to do, I think, a better job, and there is a lot of room for improvement to understand why at a certain point prostate cancer becomes castration resistant, and what we can do here to try to slow down this process and optimally treat our patients. So this is a very basic oncology slide, and it's like just I consider the Bible, if you say, if you allow me, of oncology, of molecular oncology, because it summarized the hallmarks of cancer. And here it's like just an example of how the field has evolved very, very quickly from 2000, when the original hallmarks of cancer were presented, until 2011. And there is a new one upcoming. In terms of what we now consider the molecular mechanisms that are important for cancer to be established, but also to progress. So in the new version, there is immune environment, which is extremely important for the immunotherapy drugs that we are now we can now use. There is metabolism. There's another aspect of prostate cancer extremely interesting, because prostate cancer has a completely different metabolism compared to other cancers. And then there are all these DNA repair genes, again, extremely important, because we have drugs now for patients who have this kind of genetic alteration. And then there are all the growth factors that are important for prostate cancer, but also for other cancer, and receptors. So just to give you an idea of how complex the molecular aspect of cancer is. So in prostate cancer, we have one main character, and we have been targeting this main character for many, many years. And this is the androgen receptor. The androgen receptor is overexpressed, so it's amplified in prostate cancer. And that's why the hormone therapy that we use for treatment works very well in patients with prostate cancer, because the tumor cells are driven by prostate cancer. So the androgen receptor is a receptor, and it's located inside the cells. So this is the membrane of the tumor cells, and this is the androgen receptor. So it's inside the cells, and it physiologically and also in tumor cells interacts with testosterone. So this yellow dot here is the testosterone. When the testosterone interacts with the receptor, the androgen receptor, goes inside the nucleus. So this is the nuclear membrane. And in the nucleus where the DNA is, the androgen receptor acts as transcription factor, which is its job, and increase the transcription of a lot of genes that are involved in tumor proliferation, tumor survival, angiogenesis. And one of these genes is PSA. So I always wanted to underline these, because this is why we check PSA in the patients with prostate cancer, because it's one of the genes that is directly controlled by the androgen receptor. So that's why it's extremely sensitive in telling us whether or not the patient is responding to the hormone therapy, and when the tumor becomes different, becomes castration resistant. And this is the structure of the androgen receptor gene, but I won't go into the detail. So how does the tumor becomes castrate resistant? That's a very good question. It's not really one mechanism. There are several mechanisms that can happen at the same time. These further complicate things. But one of the most common mechanisms of resistance is that the androgen receptor that's already very high expressed in tumor cells become even more expressed. So there is an amplification of the gene. So the gene that codifies for the androgen receptor expand and produce more copies of these androgen receptors. So even if we attack the cancer cells with the hormone therapy, that's not enough to control all these androgen receptor that's produced. Another mechanism is that the androgen receptor copy number is the same, but the androgen receptor itself changes. So in this case, we can have mutations that can reduce the affinity, so the binding between the drug and the androgen receptor. And then there are other mechanisms that are not strictly related to the androgen receptor. So they are related to other molecules. And the DNA repair gene defects are one of that. There are other molecules like P10. There are drugs also to target these on clinical trial. And then there is another one, HSP27, or heat shock protein 27, that is also upregulated when the tumor becomes castration resistant. Another very fascinating mechanism of resistance in prostate cancer is what we call the androgen receptor variant 7. And it's like just an abnormal version of the androgen receptor gene that lacks completely the part of the gene that interact with the testosterone and also with the drug. So when there is this androgen variant, simply the androgen receptor does not care anymore about the interaction with the drug or interaction with the testosterone and can work independently. And that's why the cells become resistant. And we know from studies published a few years ago that when there is this androgen receptor variant 7, the response to enzalutamide or abiraterone, which are new drugs that we use to target androgen receptor, is not that great. So this is just an example to underline how important it is to try to figure out why certain patients respond to the treatment, certain not, because we can try to understand the mechanism, and likely we will be able, for example, to target also this androgen variant and regain sensitivity to drug if we do that. So after this introduction, this is my agenda for today. So I'm gonna talk about some of the studies that I've done in the field of HSP27 inhibitors. So new drugs that target this protein. As I mentioned, this is important protein for castration resistant. And then I'm gonna talk about some immunotherapy option and homologous recombinant deficiency, so DNA repair gene defects in prostate cancer, and some of the trials that we are planning to do in Canada and in the US. So this is a paper that I published in 2020 and was the project I was working during my postdoctoral fellowship here in Vancouver. And during my time as postdoctoral fellow, we screened several compounds, tried to find a better drug to target this protein, heat shock protein 27, that's involved in castration resistance. And we did discover a drug. The other very fascinating things that came out from this project is that we were able to crystallize. So this is the crystal structure of heat shock protein 27. So that was not known. It was something that we did during this project. And the structure of this protein is extremely complex. So there is a whole, and there are like 24 subunits around this whole. So this makes the protein extremely dynamic. And this is why it's very hard to target and to inhibit this protein. So using this new information, we did what we call in silico modeling. So with the structure and a library of chemicals, we target the protein, try to understand if we had a good inhibitor, and we essentially identified this drug that's an old drug. It's called ivermectin. It's an antiparasitic drug, but it does affect and interact and inhibit also HSP27. So what we did in the prostate cancer cells and also in mice that were injected with prostate cancer, human prostate cancer cells, we treat the cells and we treat the mice with ivermectin, and then we check the tumor volume in the mice and PSA in the blood of the mice. And ivermectin did demonstrated to affect tumor proliferation. And also what we observed with this drug is that when we combined with enzalutamide, there was a reduction of the androgen receptor protein expression and also of the PSA. So it seemed to work in our model of prostate cancer. So what we did, and using my double skill as medical oncologist and research, I did was to try to design a clinical trial with this new drug that we discovered in our lab for patients with prostate cancer. So I did design this phase 1 trial. So there are 3 phases in clinical development of a new drug. The first one is phase 1. It means that we have to treat some patients to define how toxic the drug is and to better define the best dose to be used subsequently. So this was a phase 1 clinical trial of ivermectin with alone for toxicity assessment, and then in combination with docetaxel, or in patients with lung cancer, because the drug also worked in lung cancer, mainly affecting the epidermal growth factor receptor, which is another growth factor receptor, with some other drugs. So this is an example again of how fluid can be the discover of drug and the clinical application. And if you have like in place all the infrastructure, that could be obviously much easier. In terms of this project, we are still trying to make the drug better in terms of stability. So when you discover a drug that seems to work, doesn't mean that that's the perfect drug, but there is a room for implementation. This is what happened for enzalutamide, for example. So it was not very soluble, so they had to change the structure. So this is what we are doing with ivermectin. So we are trying to select ivermectin analogs, which are similar to ivermectin, but better in terms of stability. And there are some candidates that we are studying right now. So this is what I did with ivermectin and the future studies ongoing for ivermectin. But now I want to focus on immune modulation and DNA repair gene defects in prostate cancer, which is something new and very exciting. So why do we talk about DNA repair genes in patients with prostate cancer? This is the original publication of one of the most important papers in prostate cancer biology and genomics. It was published in 2015. It's essentially an extensive review of which genes are abnormal or are altered in patients with castration-resistant prostate cancer. And this specific figure is referred to the DNA repair genes. So these genes are important because the proteins that are codified by these genes are involved in the repair of the DNA. So why do we need in our cells a system to repair the DNA? It's extremely important because our DNA is continuously exposed to chemicals or to even like physiologic products that can alter the sequence of the DNA. And if that happens, it's very dangerous because that can lead to mutations in our DNA, and that can lead itself to development of cancer. So evolutionary, our body, our cells have learned how to prevent that to happen. So because we know, and the cells know that this happens like normally, there is there are several actually systems that control that DNA, when one cell replicates to another one, is maintained in the same sequence. So all these proteins are involved in maintaining the sequence of the DNA unaltered. And if there is an alteration that is extremely severe, the cell just activates cell death. So the cell die. That is how important it is to preserve the DNA, okay? So if there is an alteration cannot be repaired, the cell just activate cell death. That's called apoptosis. So it's an extremely preserved, controlled system. So when there are alterations in these genes that are important to preserve the DNA, obviously very bad things happen in the cells. So there are more mutation, more mistake-prone systems activated, and all of these lead to more malignant and aggressive tumor phenotypes. So what this paper showed is that about 22.7% of patients with, in these cases, it was done in tissue, but we know that now between germline and somatic mutation, around 30% of patients with metastatic CRPC have one of these genes involved in DNA repair altered. Okay, so 1 in 3 patients, which is not a low percentage of patients. So when this happens, we don't really know as yet if this can affect the androgen receptor pathway and the response of our patients to hormone therapy. It's still unclear. And also when this happens, we know that the cancer can respond to some new drugs. So this is why this is also considered predictive biomarker response. And this is why this is clinically actionable, because we can actually use new drug to target the cells, to treat the cells that had this alteration. And this is the trial that was published late 2020 in the New England Journal of Medicine with this new drug that's called olaparib versus standard of care. There was either enzalutamide or abiraterone in patients with metastatic castration-resistant prostate cancer. And this is the progression-free survivals, the probability of not having progression on drug in blue compared to the control. So the olaparib was much better in controlling the cancer in these patients compared to the control group. So based on this trial, it was a phase 3 randomized controlled trial where we participated. So Dr. Chi, who is one of the medical oncologists working here, is one of the co-authors. So this trial was what demonstrated that this drug is very active in the patients with DNA repair genes with metastatic castration-resistant prostate cancer, and led to the approval of this drug in prostate cancer by FDA and by Health Canada. So it is now available, olaparib is now available in British Columbia through a compassionate access program. So it's not funded yet, but the company is giving it for free to bridge the trial, positive results, and the funds. And it's available for patients with mCRPC and DNA repair gene defects. So how do we check for these DNA repair gene defects? Through the GU Provincial Biobank, the research program that I presented right at the beginning. So our patients are seen in all the cancer agencies across the British Columbia province, have access to this research program. They will get blood tested for germline mutation. And then in patients with mCRPC, we can also take the blood to analyze the circulating tumor DNA. If we don't have enough circulating tumor DNA, we can look at the tissue that is also part of the biobank and see whether or not a patient has this DNA repair defect. And if that's the case, we can then use olaparib. So this probably, I don't know if many of you are familiar with immunotherapy. So there has been a revolution in terms of cancer therapy with new immunotherapy drugs in melanoma, for example, so particularly aggressive skin cancer, in lung cancer, in kidney cancer, not really in prostate cancer. So there have been like several phase 2 and phase 3 clinical trials in unselected patients with prostate cancer were treated with these immunotherapy drugs. None of them actually demonstrated that immunotherapy is working in prostate cancer. And then after this failure, we did realize that there is a biological rationale to explain why this immunotherapy doesn't work in prostate cancer. And this is because prostate cancer is considered a cold immune cancer. So it means that there is not really a lot of cells, immune cells around prostate cancer. If you compare tumor from prostate versus tumor from melanoma, for example, or kidney cancer, you can realize immediately that melanoma and kidney cancer are extremely enriched of lymphocytes, so of immune cells, whereas prostate cancer is not. So one of the explanation of why these drugs do not work is simply because prostate cancer does not attract enough immune cells. And the immune cells need to be very close to the tumor cells in order for those drugs to work. So the drugs that we are now currently using as immunotherapy drugs in other cancers are drugs that target these molecules like PD-1, PD-L1, CTLA-4. So they are all molecules that try to slow down the immune system against the tumor cells. So with this drug, we just block these molecules, and then the immune cells are more active to recognize the tumor cells. So we also know that some immunotherapy drugs are associated and seems to work better if there is genome instability. So if the DNA repair genes do not work very well, in some cases we can have a lot of mutation that can lead to this genome instability. And in this case, we know that immunotherapy works better. So is it possible that there are some patients with prostate cancer and DNA repair defects that have more genome instability and therefore can respond better to immune therapy? The answer is we don't really know, and this is what I'm focusing on currently in my lab. So in this study that was presented at ASCO-GU in 2019, I analyzed some of the tumor prostate cancer tissue available in the context of our GeoBiobank for some of these markers. And PD-L1 is one of the markers I mentioned that's associated with immunotherapy response. So what I noticed in this paper was that when the tumor becomes more advanced and more aggressive, the PD-L1 expression increased compared when the tumor was right at the beginning. So in benign prostate and adenocarcinoma and CRPC and NEPSIS, neuroendocrine prostate cancer. And what was even more fascinating was that this PD-L1 expression, so the molecule that is targeted by the immunotherapy drugs, was overexpressed, especially in patients who were harboring these DNA repair gene defects. So is it possible that actually those patients can respond to the immunotherapy compared to other patients with prostate cancer who actually do not? And this led to a bigger project. So in this project, I analyzed the 500 tumor samples from patients with prostate cancer, and I analyzed the tumor cells and the tumor infiltrating lymphocytes, so the immune cells inside the tumor. So the first thing that we observed was that the tumor infiltrates, so the lymphocytes, CD8, were more predominant in the patients with DNA repair gene defects. So there was an enrichment of these cells if the DNA repair genes were abnormal. The other really interesting things was that there were other receptors, other molecules involved in the immune modulation that were overexpressed. So they were highly expressed in tumor cells, but also on the lymphocytes in the patients with this genomic alteration in the DNA repair gene defects. And those are all unpublished data. So our paper has not been submitted yet. It will be probably submitted by the end of this month. But I will just like drive you through this complex graph. So you have to look at these patients' population, okay? So the BRCA population. And here on this row, we have the tumor cells, and here we have the expression of these molecules in the peri-tumor cells, so in the stroma. So if you compare this graph with this one in patients with untreated prostate cancer or patients treated with neoadjuvant prostate cancer, CRPC, the shape of this graph and of this graph, so in patients with DNA repair gene defects, is different from the shape of this other cancer. And it's different because we have an overexpression. So this graph pointed out to this molecule that's called ARA2A, which is adenosine receptor 2A. So it's a new receptor that's involved in the immune modulation and seems to be overexpressed, at least in the patients with DNA repair gene defects compared to other patients with prostate cancer. So when we analyzed this ARA2 in the BRCA positive, again, there was a higher expression in these patients' population compared to other. And also PD-L2, which is another molecule involved in immune modulation, was higher expressed in BRCA2. But what was like really clear was the association between these two. So DNA repair gene defects and the ARA2. And this graph shows essentially what I mentioned, that the CD8, so the lymphocytes, were more abundant in the BRCA positive patients compared to all the other patients that were analyzed. Here in black, like in brown, you can see the lymphocytes. So the blue are the prostate cancer cells, and the brown are the lymphocytes. And as I mentioned, usually we don't see lymphocytes inside prostate cancer, but in these patients with the DNA repair gene defects, there was plenty of lymphocytes. So it seems that there is a correlation between genomic abnormalities in these DNA repair genes and the immune modulation. So when we quantified the lymphocyte, this is what we saw in BRCA positive, was higher prevalence of lymphocytes compared to the other tissue that were analyzed. And then we analyzed this new receptor, this adenosine receptor 2A, in the tumor cells, and again the lymphocytes inside the tumor. And we observed again, like in brown, you can see a very high expression of this receptor in the tumor cells, but also in the lymphocytes in the BRCA mutant. So the pictures are referred to the BRCA mutants, to the DNA repair gene defects tumor tissue. So again, it's a lot of basic science, but it's extremely translatable, because if we can prove that the DNA repair gene defects are associated with this more active immune environment, we could actually propose to do a clinical trial to treat those patients with immunotherapy. And again, as I mentioned, here in Vancouver, this is what we do. So we do translate our findings into clinical trials. And one of the project I'm working with, and for which I was awarded as Prostate Cancer Foundation Young Investigator, is this trial. It's called GANS, genomic umbrella neoadjuvant study. So the PI of this study is Dr. Glebe. I'm one of the co-investigators, but I wrote the sub-protocols for this trial. So the rationale of this trial is essentially that in patients with prostate cancer, localized prostate cancer, so patients who can still be cured with prostatectomy, with surgery, we know that there are a subset of patients who are going to respond better to hormone therapy, subset of patients are not going to respond to the hormone therapy. And we also know that for particularly high risk and very high risk localized prostate cancer, there is high chances that the cancer can come back. So, and these obviously reduce the chances of being cured. So based on what we know about the heterogeneity of prostate cancer, we designed this study to see whether or not we can use genomic alterations to select our patients for treatment to be received prior to the radical prostatectomy, and see if the combination of these drugs with hormone therapy can actually increase the killing of the tumor cells and therefore improve the outcomes, so improve the survival and the cure rate of patients with localized prostate cancer. So this is the protocol. It's been Health Canada approved, RAB approved by UBC and by the Central RAB of Ontario. So it's a Canadian trial. It's an investigator-initiated trial. So it was initiated by us, by Dr. Glebe, Dr. Fleschner, and myself. And the sponsor is UBC with the support of Johnson. So it's a genomic umbrella trial. So it means that all the patients eligible for this study will be screened with the tissue that is collected through the prostate biopsy. So right at the beginning. So the tissue from the prostate biopsy will be sequenced. So we'll have genomic analysis. And based on what we find in the DNA of the prostate cancer, the patients will be allocated to, we have 4 for now, but we plan to open another group. So to 4 groups of treatment. So the patients with androgen receptor driven tumor, so for example, the one I showed you with very high copy number of androgen receptor will be allocated to group 1 to be treated with maximum androgen deprivation therapy. The patients without these androgen receptor alteration, but bad actors like these genes, will be treated with androgen deprivation therapy and chemo. The patients with DNA repair gene defects will be treated with androgen deprivation therapy and a PARP inhibitor, niraparib, which is similar to olaparib. And the patients with this microsatellite instability, so a lot of mutation in the DNA, will be treated with immunotherapy. So this will be done for 6 months, and then the patient will go through the prostatectomy. And then once the prostate is out, we can actually look at how good this treatment was in terms of killing the tumor cells. So this is extremely exciting because no one had done it yet in the world. So we will be the first to do something similar. And what is also extremely important and exciting about this study is that there is an enormous translational studies opportunities. It means that we can collect blood and tissue at the beginning. We'll have access to DNA and RNA sequencing because it's part of the study. But also after the treatment, we will have access to the residual tumor. So we can actually study that tumor and understand why some patients will responded better and some patients did not respond. So and the patients who did not respond are the most informative, because we can try to understand if there are other molecules that can be targeted. So we can eventually go back and try to improve the treatment for the patients. So this is where we are at with this study. So it's going to be probably activated by September. 2021, and it's a collaboration between the BC Cancer and the Vancouver Prostate Centre. So I'll be going to the Prostate Centre to see most of these patients that will be enrolled in this trial, and then the patients will be treated with surgery by Dr. Gleave or the other urologist in Vancouver for the radical prostatectomy. So again, like just to underline how important this osmotic process between bench and bedside, and how important it is to improve the treatment for our patients. So I hope that what we are doing in Vancouver will be ultimately meaningful for patients with prostate cancer. So those are all the patients I'm working with and all the funding agencies that are supporting my research projects here. And this is my last slide. I know there was a lot of information.
Thank you, thank you, thank you. Questions, everybody? I got some questions. People you can take your mutes off. Unmute yourselves. We have two Johns on today, John Day and John Warner.
I have a question. Can you go back to the funding there? Because that's what.
Yeah.
During your presentation, I kept on thinking, okay, now who's funding this? So can you go back to this
Yes.
previous slide with the I guess it was the last one.
Yes.
With the funding. I'm just curious.
Yeah.
I always like to know about the money aspect.
Yeah, that's very important actually. So did you see my slide?
Yes.
That's the one, yeah.
This one?
So I don't see, it's interesting, I don't see the Canadian Cancer Society there at all.
Yeah, I'm not being funded by that. So I am a young investigator of Prostate Cancer Foundation. So Prostate Cancer Foundation is one of my most important funding agency. Mm-hmm. And it's a US-based, but it's an international competition. The Michael Smith Foundation for Health Research also, I got the Health Professional Investigator Award. So it's supporting my research and my lab. The CCTG and SWOG are supporting some of my projects, not immediately related to prostate cancer. And then there is the Vancouver Prostate Centre, and there is Johnson that is sponsoring the clinical trial. So those are my sponsoring agency. No, the Canadian Cancer Society is not one of the sponsor of these projects, unfortunately.
Mm-hmm. What's the GUM? Oh, I can't quite see it. Yes, that one.
GUMOC. Yeah, it's the Genitourinary Medical Oncology Canada group.
Okay.
They do award a small amount of money every year, but I got it and my fellow got it as well. So it was not a huge amount of money, but it was good as to fund some of these seed mm-hmm. projects.
Well, it's good to see a broad spectrum of agencies supporting you, so that in very practical terms, if somebody pulls out, decides that they don't want to fund you any longer, you still have other support.
Yes, exactly. Yeah, that's true. That's very important to mm-hmm. diversify your funding agency. Mm-hmm. Yeah.
Frank, you had a question?
Yes, I was just on the GUNS study. It sounds like it's only brand new patients that would be part of that, or?
Correct. Yeah, so only newly diagnosed prostate cancer patients with localized disease. So it means just right at the beginning of the diagnosis, when the tumor is localized into the prostate, and after the biopsy. Yeah, so newly diagnosed.
So the study is going to run 10 years or so. Sounds like, or 20 years?
No, because it's not just Vancouver. So it's the sixth biggest center in Canada and in the US. So there are 10 centers.
Okay, it's just with prostate cancer, it seems that it takes a long time for most patients to actually decide whether or not they're in trouble or not. And I think you're going with the prostate biopsy, probably prostatectomy first.
No, so it's a new adjuvant study. So the patient has the diagnosis and the biopsy, and then after the biopsy, the patient is started on this treatment only for 6 months. And then after these 6 months, there is the prostatectomy. So it's relatively short.
So
So
each
you're going to
patient,
see if these new treatments work
Yes,
on that specific problem for 6 months.
yes,
Then
only
you'd
6
end up
months.
getting some
Yeah, yeah.
So, and you mentioned blood biopsies.
Mm-hmm.
Or is there going to be another phase of your study where you start using blood biopsies?
Yeah.
Which treatment?
So in the GU biobank, we already collect blood for liquid biopsy. So we extract the circulating tumor DNA from the prostate cancer that is circulating in the blood. And in the GANs, we also plan to collect blood right at the beginning, so prior to starting treatment and after treatment.
so that would be after the prostatectomy?
No, prior, because you still need cancer.
Oh, so I had a question. It has to do with the criteria for eligibility into this trial.
Mm-hmm.
What would be the Gleason score and PSA reading?
Yeah. So the patients have to be a high risk or very high risk. So PSA 10 or higher, Gleason score 7 or higher, and T3, T4.
Any other questions?
Yes, I would. Eventually, the hope is that in localized, there is enough healing that radical prostatectomy doesn't have to proceed, or am I too hopeful?
Yeah, so eventually we aim to do that. So right now there is no treatment upfront. So prior to the radical prostatectomy that works well in prostate cancer.
Yeah.
In other cancer, for example, in breast cancer, if you give chemotherapy prior to the mastectomy, you can have in 30 to 40% completely disappearance of the cancer, okay? So, and in fact, they are trying to avoid the surgery for that patient.
Okay.
In prostate cancer, what we can get is at best 8% of complete response. So it's not enough. With this trial, we want to see if we do a better job by selecting our patients with DNA analysis.
Yep.
And giving like specific drugs, if we can improve that number to that 40 or 50%. So you're right, if we do see that, it is also possible that we will never need to do a radical prostatectomy for those patients who actually respond that well to the treatment. But right now, we don't have anything in prostate cancer that works that well.
If you manage to do that, you will be one of the most special people around.
Yeah.
And we want to keep you in Vancouver.
Thank you.
You're welcome.
Other questions? I think this is really exciting news.
So for my understanding is then basically your part of it is a 6-month study. See how it responds. Then the patient would go on to standard treatment.
On the prostatectomy, yeah.
Unless your treatment happens to really work well for that particular patient.
So even if it does, like we cannot really decide not to do the surgery because we don't know yet if that's enough. So that will require other further studies.
So it's not going to be that soon that we will have that information.
Yeah, so after this study is complete, we will have some information. So for example, let's see if we combine immunotherapy with hormone therapy in these patients with MSI with very high genomic instability. And we see 80% of patients with no cancer after these 6 months. that will trigger another study larger to actually validate this, because we cannot really propose this strategy only on the base of a small study. This is a phase 2 study, so there will be not a lot of patients on. So it's what we call the hypothesis generating. But if we see something so dramatic, obviously that has to be validated in larger numbers, and there is very high chances that that is true. But again, like, we cannot change our practice on the base of small number. We need like larger numbers.
So when are you starting to recruit for the study?
September, maybe. Like, we are aiming for September, because all the bureaucracy part is done. So we have just to activate the study in Vancouver, and so Vancouver and Toronto will be the first centers to be active. And then there is a center in Hamilton, Ontario, one in Montreal. And in the US, we have Boston, Dana-Farber, so Harvard University, Chicago, USD, Seattle, San Francisco, and I'm probably missing something else, but yeah.
How many people are you anticipating to recruit?
So in each arm has a two-stage design. So it means that if we see preliminary activity enrolling the first 20 patients in each arm, we will expand the group to 60. So in each arm will be 60 patients, more or less.
That's per center, or is it all for all the centers you're doing the research in?
So 60 per arm in all the centers together.
So that really becomes a rather small number for each center.
Correct. Yeah.
Maybe half a dozen.
Yeah, I think that we probably enroll in Vancouver 10 to 12 patients per arm.
Be probably the same elsewhere.
Yeah, correct.
Other questions? This is exciting.
This is Dennis from Kelowna. I just wondered if the exclusion criteria contains those that have been taken Avodart for a longer period of time.
Oh, yes.
Because there's a Canada-wide settlement on at the moment, and there's a lot of research that have been done through clinical trials and other individuals that have reported that there is some statistical significance between high-risk Gleason 8 and 9 and 10 cancer and the long-term use of Avodart. So I just wondered if that was included in your trials or not.
No, it's not I don't think it's an exclusion criteria.
Okay, thank you. Because there seems to be, a lot of attention paid to fractional amounts of PSA, 0.1 and 0.2, but then they totally ignore the use of Avodart, which is 2 and now recently 2.5 by Johns Hopkins. To say if you've taken Avodart for more than 7 years, the normalizing factor goes up considerably. So you could have a Gleason, you could have a PSA of 8 and you've been on Avodart and your real PSA is in over 20. So
I know what you mean. Yeah, it does affect the PSA, but it doesn't affect the Gleason score, right? So you can still be eligible if the Gleason score is high enough.
Okay, thank you. And as far as CTCs, I've been working on trying to get a circulating tumor cell test as a baseline for myself, because I'm big on baselining. I did request an MRI, which was refused. I requested a lot of other tests as baselines, and now the only one that I am interested in having is my circulating tumor cell test, but I can't seem to get any movement forward whatsoever on that. It seems to be a great obsession about it's only used for drug evaluation. Well, excuse me, in shared decision making, the more information, the better. And, it would help me characterize the nature.
So the CTC in prostate cancer is considered a prognostic factor. So it means that you have more than 5 circulating tumor cells I think that was the cutoff. Your
For 7.5, 5 and 7.5 have been used, yeah.
So the prognosis is worse, but it's not really used to assess. It's not been validated. Sorry, it's not been validated to assess the response to a treatment. It's always part of some exploratory analysis in clinical trials. For example, if you look at the Joan de Bono PARP inhibitor trial, the first with olaparib that was published in 2015, and also the one published in 2017, they always also include CTC as a further experimental way to assess whether or not there is a response to drug, but it's not been validated for that purpose. So it's still extremely experimental.
Yes. How about lymphocytes now? Since my radiation treatment, my lymphocytes have fallen well below the bottom threshold level, not even half of what's normal. So if we are looking at lymphocytes, would the baseline lymphocyte be a good place to start?
Yeah, so the circulating lymphocytes, after radiation or chemotherapy, there is a reduction because obviously the radiation, the chemotherapy can affect the bone marrow. And the bone marrow is where the lymphocytes are produced. So that's why there is a reduction. So having said that, there is not really any correlation between the level of lymphocytes in the blood, so the one that we usually normally evaluate with our lab work, and the number of lymphocytes inside the tumor. So in other words, you can have low lymphocytes in your bloodstream, but very high number of lymphocytes in your tumor, for example, in patients with kidney cancer or melanoma. Or on the contrary, you can have very high lymphocytes in the bloodstream, but not enough in the tumor, which is what we see, for example, in patients with prostate cancer. So there's not really a direct correlation between the number of lymphocytes in the blood and the number of lymphocytes in the tumor.
Thank you. I've been looking for those answers for about 3 years now. Thank you very much.
You're welcome.
Any other questions?
Just this quick question. Who's the lead person in Montreal that's looking after this research that's going to be rolled out in September?
You know, I don't remember the name of he's a urologist, but I don't remember on the top of my head his name. It's not Fred Side.
No, okay.
No.
Yeah. Okay.
Just shoot me a note, John, and I'll ask Lucia after the meeting.
Okay. Okay.
Do you have a hematologist on your team?
Hematologist?
Yes.
You mean like a research wise, or?
Well, the reason I ask is I look at a basket of biomarkers. I've reviewed the current literature in the last 5 to 10 years, and there's been a lot of papers written on other fractions like hemoglobin, albumin, lymphocytes, neutrophils, FSH, PSA, testosterone, put in a basket. So what I've done with my blood work is I'm looking at the trends and following the trends over time, because I'm concerned with getting the maximum warning, because I was an asymptomatic patient when I was first diagnosed. So the more time, the better. And I guess my question back to the medical community and the research community is, how come we're not doing more research on fractions, blood fractions, in terms of possibility of helping us with a prognostic or diagnostic on prostate cancer?
So it's a good question. There are some biomarkers that have been studied in prostate cancer to be coupled with the PSA. So one issue with prostate cancer is that we have PSA that it's really a very good biomarker in most of the patients with prostate cancer. Obviously, there is still like a small portion of patients with low PSA, low secreting PSA. But what I know is that there are efforts and studies ongoing to validate more biomarkers. One class of biomarkers extremely promising are microRNAs. So it's like a signature of microRNAs that can be tested in the urine. of patients with prostate cancer, and the PI of this effort is in Toronto, so it's a Canadian study. The results were presented at ASCO 2020, and the results were extremely compelling, very, very good. So what I know is that they are trying to, again, like once you study and you find something that's very promising in a relatively small number of patients, you have to validate it prospectively, your biomarker, before we can actually use it into clinical practice.
Mhm.
So they are doing the validating study now.
Okay, thank you very much.
Frank, I think you had a
Oh, I
question.
was going to go back to that, the ivermectin,
Yeah.
since it's commonly available. And what is it that it treats? It because I remember your beautiful crystal structure there.
Yeah.
That's great visualization of what's really going on. So what is it that it was good for?
So ivermectin, you mean like the prior indication of ivermectin?
I think it was a DNA problem that it seemed to help.
Oh no, the target of ivermectin in our study is heat shock protein 27. Heat shock protein 27 is the target of ivermectin. So ivermectin interacts with heat shock protein 27, and heat shock protein 27 is important for androgen receptor functions. So if we target heat shock protein 27, we also target the androgen receptor.
Interesting.
Thank you. Thank you. Thank you. Thank you. No more questions?
I'm not going to go out and start taking tons of it.
It's one of the issue we had with the ivermectin, and that's why we are trying to improve the chemical structure, is that at high concentration, it can be neurotoxic. So I don't really advise to do so, because it can pass the blood-brain barrier and can actually induce neurotoxicity. So tremors, seizures. That's why we are trying to modify the structure to reduce this transition to the blood-brain barrier. We have some good results, but it takes like very, very long to, when you reformulate drug, to actually do all the studies for chemical stabilities, metabolism, and then toxicity.
Oh, wow.
So as it is right now, I wouldn't suggest to take it at high dose because it can create side effects.
Sure.
Thank you, thank you, thank you. This is very promising information for us. It's really great to have you on do a presentation for us. Thank you very much, Lucia.
Thank you.
And if there's not any other questions, we're going to go into a steering committee meeting following this session.
Okay.
Anybody wants to stick around for that, they're certainly welcome to do so.
I have my clinic starting in 15 minutes, so I have to go.
You certainly have.
Yeah.
So, on behalf of
Thank
all,
you, Lucia.
thank
Thank you.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye-bye.
Bye now.